guys, although Stephen wasn't involved in that. We might also call this seminar by a different title. It is also looking at the impact of lateral gene transfer on community function. But I give that as really a subtitle because this is an approach by which one might make sense of communities. I borrowed this, I think it's from Andrew Hendry, his book, Eco-Evolutionary Dynamics. And this is a popular way of thinking about communities. You know, again, how do we think about communities in a way to make sense of them? And just to give you a sense, too, if we're thinking about microbial communities, let's say, you know, it's all the rage to think about the gut microbiome, the microbiome of this, that, but any of these communities are comprised of hundreds of different microbial genera, full of phages and various other selfish genetic elements. Then there's all the non-prokaryotic component, the eukaryotes as well. They're contributing presumably to some kind of collective function, even if they don't intend to. Those communities are comprised of, you know, numerous different genes that are packaged inside of cells. And we tend to think, of course, as those genes as belonging in these packages, but as Joshua points out with phages, it's clear that, you know, boundaries defined by cells, you know, don't mean much to genes. But anyway, we've got genes in there. I'm just looking for my, there we go. And those genes, of course, contribute toward phenotypes. And phenotypes give populations significance in the face of selection and other evolutionary forces, which affects the shape of populations, which affects the function of communities, and so on up to ecosystems. And there are feedbacks at every level, feeding back to every other level. So, you know, again, if you've never thought of a microbial community, the challenges of understanding them are immense. So how do people go about studying them? Well, okay. So a lot of work, most of the work these days has been largely descriptive, describe the members. And if you ever go to a microbial ecology meeting, good luck. And we can measure some aspect of function. We can measure the dynamics, actually, once we've described. So typically, this is 16S-based sequencing. Metagenomes is typically not even sequencing the entire DNA. It's just simply amplifying 16S. And using that as an indicator, it gives you a rough idea of what genera, but forget anything more than that, genera these organisms come from. One can, you know, follow those through time. It's challenging just to even do that. It's also pretty expensive. A route, too, is to perturb a system and then watch its recovery, often, though, just using very simple descriptors of that community. Some progress comes from isolating different members of the community and studying them in isolation, so one simplifies the community. But of course, although simplification is a great way to go, if you really want to understand this sort of complexity, it's encapsulated here, taking components out of context, you know, it's a game changer. You've changed the game. But you can study the interactions between different types and learn something about that. The question will always be, you know, how does that play out in the context of this community? Through isolation, indeed, one can study interactions, but it would again be nice to be able to do it in this real-world scenario. And of course, there is theory and there is modeling. There are some approaches, but the need to describe the dynamics of what's going on in this is, Joshua, open, right? There is really not much that's gone on there that captures anything like reality. And I'm not even sure, maybe in the discussion section, you could offer some perspectives on possible ways forward. But it's a problem. So what would be nice to be able to do is to manipulate one of these levels in a way that doesn't involve massive perturbation of the system, and to measure the response. The only real opportunity to do that, I think, is at the level of genes, but that would not be a bad thing. If we were to manipulate this level here, we could potentially measure responses right through the system. I mean, we could even ask simple questions, as to whether, if we were to manipulate, let's say, perturbed genes, without perturbing much of the rest of the community, could we even measure some sort of impact on the community? Or more generally, by manipulating genes, could we in fact manipulate an evolutionary process and measure its response? So I think we can. Well, the idea was that we could manipulate the flow of genes in a relatively non-invasive way by taking advantage of known aspects of the life of selfish genetic elements, which includes, of course, our bacteriophages, but additionally plasmids, integrative and conjugative elements that are kind of a hybrid between a phage and a conjugative plasmid, transposons, I don't mind what you want to call them, but collectively they're selfish genetic elements. And they significantly contribute toward the movement of DNA. They exist, of course, in our communities. Now, what we know from theory and experiment, actually, is the life of these selfish genetic elements. Well, they do have a life, and for them to maintain their living state, they need to move horizontally from time to time. If horizontal transmission is denied, it's just not possible, or it's experimentally denied, then selfish genetic elements will ultimately become one with the chromosome. They will lose their capacity to move. So our genomes, for example, are full of dead selfish genetic elements, transposable elements, once upon a time non-autonomous elements that have lost their capacity to move. Had they been able to transfer to another species, for example, before they lost that capacity to move, then they would be invigorated. Life for them would be fueled again. So environments excuse me, that limit the possibility of movement to new hosts constrain the movement of selfish genetic elements and therefore constrain the movement of DNA, of genes. But environments that promote the possibility of movement to new hosts, that promote lateral gene transfer, horizontal gene transfer, fuel the life of selfish genetic elements. So we figured that we could carry out a manipulation, and I'll show you here in this slide, that takes a complex community, that manipulates the genetic content of these communities by controlling the extent of lateral gene transfer in a very simple way. So here is our community. This is indicating a community of microbes, and although I indicate this circle here with the symbol phage, I'm thinking about all selfish genetic elements. I'm lumping them all up here. And there is a constant toing and froing. I don't know who said it, but I certainly believe it after what I'm going to show you today, is that genes that matter, genes that are ecologically relevant, will be on the mobile gene pool. Everything else is dead. So one understands an interaction between genes on the mobile gene pool and community members. And although we've got borders around here, again, there's a sort of a continual flow. Our ability to think about and even portray this sort of interaction is not well developed. So we target a compost heap in Square Chipion, just down the road from Euspisy. It's a compost heap that we've come to appreciate. It's got a lot of phage in there that attack our favorite bacteria, much has been characterized. So we went to this compost heap and we took 10 1-gram samples of compost, independent samples from this compost heap, and we placed them into 10 mesocosms. So these are small bottles that contain 20 mils of minimal salts media. So that's giving a source of nitrogen, ammonium chloride. The carbon you can't really, inside of each of these is a piece of paper, about 4 square centimeters. Paper is comprised of cellulose, and of course, compost material is a lot of cellulose in there. So we've given them a single carbon source and a very minimal environment. After establishing these communities, well, actually, let me ask you some questions. So if I told you we were to do what I just told you, which is to try and maintain a community, and I'm talking about all this complexity on a single carbon source, what would you think about that? The ecologists among you. I mean, it's, you know about Gauss's niche exclusion principle? You do? What would you expect? Single carbon source. We wouldn't expect to see much surviving there, right? But we also know that resources will be partitioned. So, cellulose, a polymer of glucose, will be broken down by some members of the community, and that can be partitioned potentially exhaustively. So we figured, well, let's take 1 gram of compost. There's going to be a huge diversity of organisms in there, and indeed there are. It's full of eukaryotes, nematodes, everything. We put it onto cellulose. We left it for 2 weeks, and we sequenced at that stage. There was still an awful lot of stuff, and after 2 weeks, we homogenized those bottles and took 1 mil out of the 20, so that's a lot. And at this stage, the paper is a bit of a slurry, and transferred it over. And we found there's a lot of microbes still, a huge diversity. I'll show you some. So what I'm saying there is we found that we could establish a very diverse community of microbes on the simplest of carbon sources, which is actually a real surprise. And we'll talk a little bit more about that later on. But here is the key aspect of the experimental design. Here is 1 replicate with our microbial community, and I'm thinking about the selfish genetic elements here. That community is split after 1 month of establishing the community into 2 sets, or 2 microcosms, mesocosms, one of which we label vertical and the other which we label horizontal, and you'll see the reasons for this in a moment. So this manipulation is one that is going to affect, we predict, the movement of genes. So here are 2 independent communities, and we have 10 that are of the vertical kind. To show you what this involves, vertical simply means taking every 2 weeks 1 mil of this population, passing it into a new microcosm. And that continued on for 48 weeks, a year. Same here, pure vertical transmission. Everything that's in here is passed on to the next generation. Of course, within here, there is a lot of dynamics involving phage and so on, but we are not implementing in this regime a horizontal or additional treatment that allows horizontal movement. So this community with the red circle is is paired with this community with the red circle. The purple circle, this is the pair. So the vertical, the horizontal, the vertical, the horizontal, and there are 10 of each. So how do we treat the horizontal? Well, as follows. We collect the phages. Well, let me say more precisely. We take a sample of this community and put it through a 0.22 micron filter. And that removes the bacteria, leaves phages, but it also leaves naked DNA, anything that passes through a 0.22 micron filter. We kind of refer to it as the phage juice. So we get phage juice from here, the juice from here, and in fact from all the 8 other populations, and we mix it together in a single flask. We then carry out the vertical transfer, but we then follow it up with a dollop, an aliquot, of this pooled mix of selfish genetic elements. Okay. So, what have we done? In the horizontal treatment, we are taking a sample of the mobile genetic elements, we are pooling it, and then we're distributing that across all of the independent replicates. That's not going on here. What we would expect here, then, Well, here, we are certainly giving life to selfish genetic elements. Things that depend on moving into new genetic backgrounds, their life is being fueled here, and it's being somewhat impeded, well, potentially significantly impeded in this treatment here. So a simple design, in principle, this should allow the manipulation of genes, the movement of genes that matter here and not here. Now, one thing that Joshua, I don't think, touched upon is that these selfish genetic elements, like bacteriophages, like plasmids, like integrative and conjugative elements, and all manner of other types, while they have a life of their own, very often they inadvertently pick up DNA from the genomes of the organisms that they find themselves in. And this gave rise to the process known as transduction when it involves phages. So, bacterial cell, host genome, in comes phage DNA. Now, if you read the textbooks, typically it needs to integrate into the genome, and then when it excises, it takes a little bit of DNA from the chromosome and packages it into the next generation of phages. Okay, well, there's a phage particle, and it contains phage DNA plus a little bit of the host genome. And so these phages can move DNA around that's not part of them, it's part of the host chromosome. Now, I think this is much more prevalent than we realize. We tend to think of this being a property of simply lysogenic phages, temperate phages, but anything that is moving around has some chance of picking up other DNA with it. Now, if that DNA happens to be useful to a bacterium that acquires that phage, for example, and it integrates, that bacterium may be delivered genes for degrading cellulose, for example. And they would be massively amplified in those populations. So by promoting the process of lateral gene transfer, we're also thinking more generally that anything that matters will be on this laterally transferred gene pool, and that should allow genes that are functionally significant to move very rapidly through these populations. So there's a prediction, but equally we could have made a prediction the other way around, that the horizontal treatment fuels the movement of genes and affects community and or even ecosystem function. So what we might expect here then, following from that prediction, is in treatments where horizontal gene transfer is rampant, these communities should evolve to degrade cellulose more rapidly than these populations here, within which lateral gene transfer has not been promoted. I don't think we really need to look at that. It makes, yes, this is what I wanted just to point at the bottom. So again, this is another representation of the experiment. In case you didn't get it, again, the vertical just goes vertically. With the horizontal treatments, they're getting the pool of phages from all treatments, and they're being distributed. Every 2 weeks, they're getting every 2 weeks. So on one hand, really allowing genes to move. That's at least what we think. And we look, we sequence a great deal. So from the outset here too, potentially a great experimental design. It's wonderful to have good ideas. Of course, the other thing that is necessary is means to get data that allows you to assess the validity of these ideas. And again, the complexity of these communities is a massive challenge, even with deep sequencing. But what we did try is simply to sequence everything in there and see if there was enough information returned to us to allow us to make sense, well, to be able to detect movement of genes, so hence a lot of sequencing, and also looks at, we've been looking at function as well. So first of all, a comment on diversity. This experiment has generated a vast amount of data. We could spend probably the next 10 years with a whole team of people just pulling it apart. And if you guys came and joined it, we could even perhaps model some of this. We really haven't done much on the diversity side at all. But this to simply show you that across 48 weeks, there is an enormous amount of diversity that is maintained on a single carbon source. This is the number of genera from one of the sequence databases. There are hundreds of genera maintained right throughout this experiment. To my amazement, when I looked down the microscope at these communities, there are even nematodes in these communities 1 year on. I'm not going to show you the data here, it's another story, but these communities have set up their own nutrient cycling regimes. Their entire nitrogen cycles operate. It turns out, well, we learned from the genes that we saw being moved that these communities are actually nitrogen starved. And indeed, there's a lot of carbon there, the minimal amount of nitrogen. And so different communities have different organisms that are fixing nitrogen, assimilating nitrogen, denitrifying, nitrifying, the whole process, complete sulfur cycles, phosphorus cycles, maintaining a diverse pool of eukaryotes as well. But you can also see from here that there really is no effect of the treatment on overall species diversity. So this simply shows you that diversity is maintained. And one thing I would like to do in the future is actually to manipulate levels of diversity by cutting the paper. Because a sense here too is that there is a whole evolutionary process going on that is specific to the community context. We know lateral gene transfer happens. We can see it with individual phages and individual bacteria. But what about at the level of a community where there's this huge pool of DNA that can be potentially moved around? Presumably there is a process involving lateral gene transfer that depends on a minimum diversity, a minimum pool of library size in order to provide opportunities to realize an impact at community level. So there is a lot of diversity there, and that's nice. You can look down the microscope, not the most elegant photo, but this is a cellulose fiber. These are eukaryotes. And very dense microbial communities, biofilms if you want, that form on the surface of these communities. There's almost no growth in the medium. It's all on the surface. For a long time before we got a microscope, I used to worry. I'd come in every day and look at these microcosms and I'd shake them and go, there's nothing in there, there's nothing growing. And then we sequenced and we saw there's an awful lot there, and then we got a microscope and I stopped worrying about density dependence because, if there is low density of hosts of bacteria, Joshua's talk, then we would not expect to see much activity on the phages. But as you can see, there is a huge density of bacteria in close proximity to these cellulose fibers. Okay, I'm also just going to whistle over this. This just shows some of the common taxa, but I don't want to particularly dwell on the diversity because we haven't taken that part apart. So comparing the metagenomes. So are DNA sequences moving around? Is this treatment actually fueling the process of lateral gene transfer? This experimental design provides various opportunities to explore that, but one comes from simply sequencing T0 and then sequencing some latter time point, vertical and horizontal. All of the reads, the sequencing reads, so we are doing this initially with just short read, shotgun sequencing, entire metagenomes. So all of the reads that we find here, fragments of the chromosomes, fragments of the DNA, come from here. This is also true here, except we would expect to find a set of unique reads in here, i.e. reads that have moved via lateral gene transfer. That's if our experimental design is working and if we have the tools to take it apart. So, of course, this allows a fairly easy pipeline just to look for these unique reads. And we're talking about, billions of reads from each microcosm. So computationally, this is extremely intensive. Well, you will see on the left-hand side of a number of these the workflow, but we don't really need to pay much attention. Just let me flick back. That's a workflow to identify these unique reads. And what we do effectively is find the unique reads. We don't assemble anything at that stage.
I'm not sure I understood why you would expect unique reads in the horizontal treatment and not on the vertical.
So, V1, H1. V2, H2, and so on. Vertical, we just transmit a serial transfer, a sample, well, a very large sample, 1/20th of what's in here comes into here with a new piece of paper. That happens here, but remember, what we additionally did, well, let me complete this. So this is all just vertical, but of course here are our horizontal treatments. What we additionally did, and we have a little flask over here, at the time of transfer, we took a sample here, put it through a 0.2 micrometer filter to remove all the bacteria, and that leaves us what I'm going to call the phage juice from horizontal 2. We do the same from here. So in here also goes phage juice from horizontal 1 up to 10. Okay, so that's got the, and what we would expect, well, we've taken the genes from all of the horizontals, put them into here, and now we've distributed them into all of the horizontal only. Too many arrows now. Okay. So every horizontal treatment received. So when we sequence, we should see, if we sequence horizontal, we should see a set of gene sequences that have come from this pool, and their origin would be from some other bottle. They've been moved by these selfish genetic elements that are in this phage juice. So simply a set of new sequences that, if they're unique, they're not in here, well, in the founding one, they've got to have come from somewhere else. And we would expect that if our experimental design has worked. So what we see, to my absolute amazement, within just 2 weeks of implementing this experimental regime, are tens of thousands of unique reads. It almost seems too good to be true. So we can go a little bit further with these reads. We can attempt to assemble them into contigs, all right? So we have reads. Each read is about 150 base pairs in length. Just to, there's a huge amount of DNA in here, in these microcosms, and we get a picture of many samples of it, 150 base pairs at a time. We've identified the unique reads, and then we can say, can we assemble them? i.e., will they overlap sufficiently to give rise to a larger piece of DNA that we can then ask, by interrogating databases, what does it look like? So given the vast amount of data, but also the vast amount of complexity in DNA in there, I was surprised to find that we could actually assemble these sequences, often into contigs, continuous pieces of DNA, up to 100 kilobases in length. That really allows us to say, well, what are these things? Well, here's a snapshot of the sorts of things that are in that unique and therefore moving around gene pool, effectively the moment that we implement this treatment. You've got things like CRISPR, right? Protection against viruses, exactly the sort of thing you would expect to be moving around. Restriction endonucleases, these are involved in degradation of foreign genetic elements. So, these are things you find on phages and plasmids. Toxin-antitoxin systems similarly. Integrases, components of phages, selfish genetic elements, genes that are predicted to belong to phages. A large class of genes here that are involved in degradation of cellulose, genes for nitrification, nitrogen fixation, and siderophores, which bind iron. Now, all of those are good candidates for things that matter. And this is quite interesting. So just 2 weeks after splitting these treatments, we see from our DNA sequences that the genes that have been moving, or the DNA that's moving horizontally, already has a lot of genes on it for degradation of cellulose. Now, to me, this gives life to the idea that anything that there is this constant flux of DNA, forget bags around cells really, DNA is moving, and a lot of it is going into this pool of elements that moves things horizontally. We apply the selective criteria, and immediately we see the presence of these amplified. So all of these are unique genes or unique functions, or functions that are in the unique gene pool. And again, after just 2 weeks. Well, we can take a closer look. So here are a set of phage genomes. I'll show you, let me flick forward. There is one phage. No, you don't need to take any notice of it other than to be amazed at the fact that out of all of this, we can even assemble complete phages. This is like so this must be extremely common. So that's one phage. Here's a set of, I think, 25 phages in the unique gene pools. And here are the bottles, H1, V1, horizontal 2, vertical 2, horizontal 3, vertical 3, the pairs, the 10 pairs. It's a heat map. The brighter the green, the more sequences there are. Yellow, fewer, gray, none. Now let's just look at this sequence here. So this sequence is found in horizontal bottle 1, but not in its vertical pair. So it probably didn't originate in the vertical bottle here. Here we are. Both the vertical and the horizontal have this phage. This phage probably came from here and is spread into the horizontal 1 bottle, into the H5 bottle, into the H6 bottle. Okay, it's also in vertical 9. So we can't be sure whether that phage was in the H2, sorry, in sample number 2 or in sample number 9. But you can see that where it appears elsewhere, it's only in the horizontal bottles. We could go through the same story, but just look down the columns, and you can see every second one tends to be much more filled in. This is an indication, or this is what you would expect, if these phages are moving from bottle to bottle, and one can trace the genealogy through, and one can also trace the dynamics through time. I'll show you in a moment. So it looks like things are moving around via our treatment, and then we can even see this, and we can even assemble entire phage genomes. There is one of them. Yeah.
What it means when you find the vertical one? There are some that are in horizontal and vertical, for instance, the third line.
Yes, so the likely explanation is that that phage was present in sample, if we look here, in sample number 2 from the get-go, in sample number 5 from the get-go, in sample number 6 from the get-go. Yeah. So it's entirely possible that one phage is in multiple independent samples right from the beginning. So if it's in a vertical bottle, like there and there, it was likely in the original sample.
So you are interested only in the transcript genes that are present only in horizontal and not in vertical.
Right. These are the ones that we see. So when you see, in horizontal bottle H1, there's 1, 2, 3, 4, these first 5 phages, none of them are in the corresponding vertical bottle, and yet they're descended from the same sample. They almost certainly came from somewhere else. And then you should be able to go back. And so here, with this one, you would say, well, because it's in both the vertical and the horizontal, it was probably in that sample, but also it looks like it's in that sample. So we couldn't be sure it came from 9 or 2, but it certainly spread into
because you mix everything.
Yeah, that's right. That's right. Of the horizontal only. That's right. Yeah. So again, one can gaze at these phages, so there really are things moving around and we can detect them. But it's not just phages. I already pointed out that we had genes involved in cellulose degradation. And here is a set of them: endoglucanases, carbohydrate binding proteins, beta-galactosidase, glucosidases, I should say. These are all implicated endoglucanases in cellulose degradation. Here is a piece of DNA that doesn't look like a phage. Maybe, well, it has some phage-like elements in it, but what it encodes in red here are genes involved in cellulose degradation. Now, this sort of approach that we've taken too, maybe there are all sorts of selfish genetic elements out there that are new to be discovered. What we know is that this piece of DNA, and again, it's about 80 kilobases long, it's a big chunk of DNA, it's in the unique sequences. It's moving around, and once again, we can track it. Here actually are some tracks of just 2 phages and 2 elements encoding cellulose degradation genes through time, through the course of the experiment. So this is rather interesting. This is a phage that was found in community 4, both horizontal and vertical. After 2 weeks, well, it's also there in V1, but after 48 weeks, this is how it spread. And again, look at that. It never makes the vertical, never makes the vertical, never makes the vertical, never makes the vertical. It's being spread by this process of lateral gene transfer, and one ever has a sense of the dynamics of this. One could quantitate this in the context of this complex community. Here is another phage. This one, as you can see, is far less successful. It spreads, but it then dies out during the course of the experiment. That shouldn't say phage. This is, yes, so these are, again, unique sequences involved in degradation of cellulose. And again, they have similar trajectories. Very successful here, spreading throughout the horizontal communities if they weren't already in the vertical. But again, you look at the vertical, never, 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 never. Vertical, yes, but it had it right from the get-go, had it from the get-go. Well, that could be contamination. It could have been just very rare here. Then it comes and it's gone. So this experimental manipulation is having the effects, and we're able to measure these.
So this would be like either an error or a contamination, because like on the vertical one, it appears on the second week, then you don't detect it for several weeks, and then on week 20, it appears again.
Right,
Vertical,
yeah. So it's possible it's contamination. It's always possible. The other, more likely, is that we have failed to detect it. I'll talk more about the limitations of our data. So things that are unique could have come from a horizontal bottle, or they could be in that original population, but very rare, and we failed to detect them. And distinguishing between those two possibilities is something that we spent a lot of time doing, and then when we rerun this experiment, we will change the design a little bit to make sure that we can get around that. So, again, many genes. So this is a set of genes that are unique even 1 year or 48 weeks on, the same sorts of genes. This is a large number of contigs that have been moved that even at the end are unique. So this shows the overall trajectory of those unique sequences. So right at the start, of course, completely there. This is the defining population. We lose sequences through time, although they can, flatten off. These sequences, these unique sequences are being maintained, move around, are being maintained. What about the function at 48 weeks of these unique genes? Well, we have here comparisons between T0, our vertical and horizontal 1 year on. They're based on predicted function, carbohydrate metabolism, amino acid, so on. This set of genes here, and these functional classes, have not changed significantly over the course of the experiment. They're same numbers in horizontal and vertical. These, though, have changed significantly, either up or down, between vertical and horizontal. So, for example, nitrogen metabolism. There is a significantly greater number of nitrogen metabolism genes in the unique gene pool 1 year on compared to vertical. It's true up here, it's true here. It's a decrease here. So here we start to find differences between, functional differences between vertical and horizontal communities. Nitrogen metabolism, so I mentioned before, we hadn't appreciated that these populations are nitrogen limited. And it seems that getting nitrogen, well, you need nitrogen to degrade cellulose. You can't degrade cellulose without nitrogen. So nitrogen seems to be the limiting factor. Here are a set of genes involved in, genes involved in the process of nitrogen fixation, denitrification, and so on. And you can see, particularly if we look at ammonia assimilation and ammonification, that there are significant increases, actually slightly higher in the horizontal than the vertical, both the same there. The genes involved in nitrogen fixation at 48 weeks are significantly different than they are at T0. We're going to continue to home in. So I mentioned before that we could have unique, so I've emphasized, the sort of unique sequences in our horizontal, so a sort of set of unique. And that's because we don't find them in the vertical or in the T0 bottle. But as I pointed out, some of those unique sequences may in fact not have come from horizontal. It's just that we failed to detect them here because they were at such low frequency. Now, there are ways around that in future incarnations of this experiment, but one thing we did is went back and just sequenced like crazy. We put these through next high-sec sequencer. This gave us something like another 2 or 5 gigabase, a huge amount more data from our T0, so we could detect rare types. And this simply shows that if we add, I think, 40 million new sequences at a time to our T0 bottles, our capacity to detect, with the exception of one bottle, which was under-sequenced earlier, really doesn't change. So we are sampling pretty well these populations. We can then ask about unique sequences in vertical, and there are some, so those are sequences that were rare to begin with, and here they are in horizontal. It's significantly different in the horizontal, but there is a fraction of unique sequences that are also unique to the vertical and therefore must be rare sequences. But even with that in mind and that taken into consideration, if we look at just the genes involved in nitrogen metabolism, there is a significant difference in the presence of these sequences in the horizontal gene pool. There really does seem to be more moving around there. If we zoom in even closer, it becomes more compelling. So red indicating the unique open reading frames involved in these nitrogen-related functions, fixation, regulation, and other nitrogen genes. And you can see in most cases, the number of unique sequences is significantly greater in the horizontal gene pool. So the suggestion is that these genes are really making a difference and that they're moving around. And in fact, by seeing this, this is telling us something about the community. So we're nearly there. The obvious thing that one would do is try and link this to the rate of cellulose degradation. And one of the reasons for choosing a piece of paper as the carbon source is that, in principle, cellulose degradation or assays for cellulase are easily done. The reality is it's not true, and the signal-to-noise ratio is just too great, and we couldn't get a signal. So then we turn to nitrogen, and we spend a lot of time developing mass spec methods for putting in labeled isotopes and looking at their rate of incorporation into biomass. And after a long time, I don't know if you've ever kept a tropical fish. If you keep tropical fish, you would have done measures of water quality, and you would have assayed ammonia, nitrate, and nitrite. These are the products of ammonification. And so we just sat down and did this. And what this shows is that if we measure across all bottles nitrate, nitrite, and ammonia, very low levels and no significant difference of these two components. But ammonia, there is significantly more ammonia in the horizontal bottles. I mentioned that the transfer regime involved 2 weeks before transfer. So what Stephen then sat down and did was to measure ammonia levels at these time intervals across 2 weeks in both our vertical communities and our horizontal communities. And you can see in every single instance, the amount of ammonia is greater in the horizontal community than in the vertical community. So there really is movement of genes that have resulted in a measurable functional impact on these communities. What does it mean? I'm not actually sure. It gets complicated here. And in fact, although we think we know a lot about nitrogen metabolism, when you go into the literature, there's not so much known. There are two possible angles on this. So the first thing, it's rather unexpected. What it means is that the process, so ammonification takes dead things and degrades the protein, produces ammonia. And ammonia is soluble and is readily taken up. It's the most easily assimilated form of nitrogen. So you would not expect to see lots of ammonia. It should be taken up immediately back into biomass. One possibility, though, is it reflects elevated metabolism on the paper. Oxygen is low, and if oxygen is low due to elevated metabolism in the horizontal communities, ammonia levels will be high. But there is another intriguing link. It comes from the farming industry. It's long been known that cattle are more productive if you put ammonia into their feed. So maybe there is something in the nutrient cycling here that has resulted in ammonia being more plentiful into the system because this is directly linked to greater biomass. In a way, it doesn't matter too much. What's super to see is that there is a measure of function that shows a significant difference between horizontal and vertical treatments. And when you consider the complexity of these environments, the fact that we have 10 independent biological samples, to see something so reproducible in every community makes me think it's real. So I started off by saying communities are complicated and how do we study it? So this idea of manipulating genes via a fairly simple manipulation where we don't have to disturb the communities seems to, I think it says, it works. We have the technology and the means to be able to detect these genes. So the experimental design is certainly allowing fueling of the process of lateral gene transfer. We have the technology and the computational power to detect it and even link it on through to function. And it also shows en route of many things here that we can even capture the dynamics of individual elements within the community. With barcoding technologies and so on, we can use this approach to do much, much more. Certainly application to complex systems, we can generate quantitative data and get insight into the eco-evolutionary dynamics that really does underpin this community function. So we've learned a lot from doing this experiment, and it's now gearing up again with some modifications. I won't go through this in the interest of time. More replication, particularly on our horizontal and vertical pairs. We only have a single replicate of each, so we can't say much about ecological changes. One would spike these communities with organisms with barcodes in the genome, therefore it's easy to recover these genomes at the end. One can measure precisely the rate and the amount of DNA that is taken up through these communities in time. So compare this with the Lenski experiment, for example, or our Pseudomonas experiments. You take a single genotype, you propagate it on its own. Give it a year, and it's done a couple of thousand generations. We sequence, we know we will find maybe 20 mutations. On cellulose, if they can degrade cellulose, they will go nowhere near that number of generations. You might expect to see 1 or 2 SNPs in a genome, single nucleotide polymorphisms, after 1 year of evolution. You put that same bug in the context of the community, and the genome is utterly peppered with DNA that's coming from outside. So the impact of the community on the evolutionary trajectory of individual cells within populations seems to be immense. It's something we really want to get a very precise quantitative measure of. One can, of course, incorporate fluorescent markers, so microscopy becomes more possible. So Romain Kozul at Institut Pasteur in Paris, he has developed some very nice, he's a biophysicist. He's used this technology called chromosome conformational capture. It's a way of capturing tertiary structure of genomes. So it looks at interactions within the genome and it captures these. And he realized you could take this technology to complex communities. This is a representation of the dynamics of genes within such communities. This here is actually a phage. These are bacteria. This set of genes are all on the phage. It's possible to understand this because they are linked physically. You capture this. But these genes are also linked via these lines here to these bacteria. What this means is this element is spending part of its life in this cell, part of its life in this cell, part of its life in this cell. So this kind of representation, and developed actually using social network theory, allows one to take an altogether more dynamic view of what's going on within these communities. Down here where you see composite colors, that's because there is a plasmid that has come into this bacterial population. The same plasmid is also over here. So one can begin to capture the dynamics of gene movement, really see what's going on. You see, genes without borders here. It's a whole incredible dynamic of stuff moving around, of which we have almost no understanding. So even my enthusiasm, I hope you might be inspired a little bit. Come and work for us. Come and work with us in a postdoc, or in which one can perhaps get a handle on some of this really cool stuff. Andy Farr, who's studying this, is adding additional treatment effects. Put some antibiotic in, for example, and one can look at the movement of these antibiotic resistance genes very easily. They will immediately be put up onto that horizontal gene pool. There's much to be done in terms of metabolism and metabolic models of community function. There are some very nice approaches developed already, but much more to be done. So with that, thank you. And again, this has been largely the work of Stephen, supported by a Marie Curie research fellowship. And we're definitely looking to expand this area of work. So if you're interested in the future, please get in touch. Thank you.
Would you prefer to take the questions?
Yeah, let's just
right
take
now?
a few questions,
Yeah,
and then let's get on with it. Yeah, if you have any questions, ask away. Otherwise, we can just have a beer.
Just, what is the time scale of degradation of a single piece of DNA compared to these 2 weeks?
So what's the rate of degradation of?
A single piece of DNA in this pond.
No idea, no idea.
So it might be that from one experiment sequencing to the other, you see the same. You see
just being maintained.
Exactly.
Without being in an organism. It's possible, but I would not say throughout multiple transfers, because each time it's being diluted down. There is such a vast amount of DNA. For us to actually detect anything in here, it has to have been amplified, I would say, a lot. Mm-hmm. That's why we can pick up phages. Presumably when it goes into a new background, finds a naive host, it is massively amplified, and and we can then capture that signal. So I think it's very unlikely that DNA would persist. Also, DNA is degraded. It's great food for bacteria. Naked DNA tends not to hang around very long in natural environments. Yeah.
The main idea of this experiment is to accelerate the exchange of DNA sectors, parts in an environment.
And to manipulate that whole evolutionary process that is driven, yes. To speed it up on one hand, and on the other hand, constrain it.
Yeah. Something that you didn't mention, there is also DNA transfer between bacteria.
Yes.
And then you didn't mention, and perhaps some little transcript that you find in the vertical may be because there is bacterial transference, maybe.
Indeed, and that will certainly be going on in our vertical treatments. We're not stopping that. It may also be when we pull this, this DNA through a 0.22 micron filter, there will likely be naked DNA in here that is naturally taken up by the process of transformation when it goes into the new vial. But yes, we are not, well, we are fueling the process of transformation, the uptake of naked DNA, by this treatment as well. And it seems, there's been a number of discoveries recently of other mechanisms by which cells seem to be able to send off DNA, rather like the blebbing that Joshua talked about, putting DNA into packages and sending it out there. All of that comes through here. And maybe there's, a lot of these elements that we've looked at that are in that unique gene pool and we can see moving around, they don't look like, they're not obviously a plasmid, they're not obviously a phage. Maybe there's whole categories of things out there that are moving around that is new.
But we can also infer that in the beginning of the life of the planet, after the bacteria and virus appears, virus is a main component to this diversity and to the development of life.
Yes, exactly. But they're not the only elements that are moving sequence, and very often it's simply inadvertent, but you can see too that if you are a selfish element, then a great way to succeed is to have picked up some DNA that is really useful to your host, and then, thinking about some of the work that Joshua presented too, you may aid your vertical dissemination by providing genes that improve the fitness of hosts. I think this must be going on here on a scale that we haven't really appreciated. The fact that we see all these ecologically significant genes on that horizontal gene pool effectively instantaneously, there must already be, this flux must be constantly there, and then we run this experiment and suddenly we see all of this stuff that is ecologically relevant in this environment. I don't think it's the alternative is nothing much is happening until we impose some selection, and then phages and all these things start doing their work and they start picking up DNA, and then we see it. We probably wouldn't see that sort of response for a long time. I think it's much more consistent with this extraordinary dynamism of genes moving around all the time, and the moment the environment changes, those genes end up wherever they happen to end up, and if they're useful, you see them amplified.
And you believe that this kind of phenomena is happening also in the ocean, but in a low level, a more diluted level?
Indeed. So it's going to be dependent on host density here, for sure. That's a major factor. But there's no, again, Joshua gave the work from with the phages in the cyanobacteria, the work of, in the bioarchive. So that shows, again, from sequencing, you can see the movement of these elements in the oceans,
So I got really puzzled about how the horizontal transfer enhances the production of ammonia.
Yeah.
So very interesting. So in some sense that you could use that as a proxy of ecosystem functioning and production.
Thank you. Yes,
And
that's right. Even though I don't really understand, but there is no doubt there is a function that we can measure that seems to have been directly affected by the treatment. Yeah.
Yeah, and so, but when you see the diversity over time between the two treatments, they are not that different.
That's at the level of genera at 16S defined levels. So that's not.
Because what I was thinking is, if the diversity is still the same, and when you have the horizontal treatment,
that's right,
this somehow increases the production of that environment or that microcosms. What does that mean if the diversity is almost the same?
Right. Well, yes. So that's diversity at the level of genus. That's all. So that really, that's there. And then we ask about. We haven't asked about diversity. We've asked about the number of it's a number of unique types in horizontal versus vertical. And there, when you focus in on different functional categories, you see more in the horizontal. So there's no reason it should be. We're looking at a very fine resolution when we're asking about unique sequences, whereas 16S is a very, very crude measure. All that's really showing is that over time, there is a large amount of diversity and it is being maintained. Okay. There's Mr. White's.
One quick question, one medium question. The quick question is, it said CRISPRs. Do you know if those were anti-CRISPRs or CRISPRs?
Don't know, but we could certainly look.
Okay,
Yeah.
just because there's a lot of interest in anti-CRISPR
Yeah.
biphage, and so
Yeah,
it might be worth
exactly.
poking around.
It would, yeah. There's so much interesting to poke around.
Yeah,
definitely, yeah.
And the other thing is that phage can kill, and yet you're thinking of them also as mobile, and they have dual roles. Maybe
Yeah.
they're killing, maybe they're moving genes.
Yeah.
Is there any way? I was thinking if you somehow UV expose the pool so that they're probably not likely to kill, but still could get in, and it's a little tricky. Have you thought about ways to accentuate the mobility side without the mortality side?
Yes, I mean, it's a good point. So again, we've sort of stepped back, as you can see here, from any of the details and tried to get at something from a sort of a community level. So one thing we haven't done, which we could do, everyone suggests we should do it, but actually it's not so easy, is to sequence directly the phage juice. The trouble is we would have to amplify it, and but we could do that. That would give us. I mean, well, it would tell us if we could do this. I mean, I guess we can cesium chloride purified. What really is in that pool of sequences. On the other hand, we really get a snapshot of that by seeing what's in the unique set. But it would be. I'm really intrigued with this sort of, this dual role of all of these elements. Of course, they're making a living for themselves, but inadvertently, I think, probably most of the time, inadvertently, they're simply picking up stuff that's around, and there's this whole dynamic, whether they're lytic phages, lysogenic phages, plasmids, I think, just shit is happening all the time, and that's what we get a snapshot of at the level of a community, But how much of it is specific to phages? How much of it's totally accidental? I don't know.
And then if the students will indulge me for one last one, which is that there's barriers to gene transfer at individual cells level. So if I diverge too much from another cell, it's very hard for me to transfer a gene. Now you have communities which could diverge.
Yeah.
And have you thought at all about community level barriers to transfer when you do these kind of mixing?
Yeah, no. Again, we've taken the sort of the approach we've taken, which is not, I guess, deliberately not worrying about the details. Let's just see if we can pick this dynamic up. It does raise many detailed questions, as you ask. When you say, there are barriers due to DNA similarity or not, that's for homologous recombination, right? So here we're probably not seeing much. Well, we do see that. We can see that via another tack on the data, but largely we focused on probably what is moving around via, horizontal gene transfer, illegitimate recombination. So it may be, we haven't looked at what hosts, but it may be stuff is moving around much more broadly than we think.
So I guess that's why it would be the same mechanism, but for example, for the plasmid of the phage in the first place, if they're too divergent, it may not be so. So I was just wondering, diversity, maybe for different reasons, the phylogenetic diversification of your H's may start to lead to some groups that can transfer.
Yeah, that's an interesting. I mean, we certainly see some of the elements that are moving around are not terribly successful long term, despite being spread. So, I mean, we could probably correlate that with the fluctuations in different genera or even species. But again, we haven't delved into that side of it yet. All right. Thank you. That's it. That's it. Thank you.
Yeah, let's stop here. And thank you. Yeah, so last activity of the day is the seminar an hour from now, 4:45.